started and who discovered it, well, that is documented already. But where we are now, we are on gene sequence. Some of your colleagues who will present today, they have worked on the viruses in a situation which was actually disturbing or attacking on the developed countries also. And our people were there for their degree programs or some activities, and they contributed. And they will be also having their papers today's conference, in today's session. So I thank Pakistan Poultry Association, that particularly Dr. Abdul Karim Bhatti, who is a great leadership with us, Dr. Asim Mahmood Khan, the chief organizer of IPEX 2022, and all of your organizers. I cannot name one by one, but everybody has a splendid role in managing or arranging all of us to be under this roof. So please, when you think, oh no, I am undergraduate, I am postgraduate, I am PhD scholar, or I am postdoctorate, never matters, you're all professionals. And we are waiting for you in the industry to come with ideas to make your space, because bacha will never find a space, because nobody wants to give responsibility to any bacha. So behave like you are professionals. And I must tell you with pride that the chairman, Pakistan Poultry Association, the chief organizer, and a list of people who are leading the poultry industry were one day on your desks, and they become leaders of the industry. So why not you? So look forward, and now once again I will request Dr. Shahid Saab, PHEC chairman, to generously support your research, because this model that the industry and the researchers are sitting together, and we have to bring ideas. Up till now, we have been buying ideas, but we have a lot of opportunities that we can create our own idea and then adopt it, and then become leading business in our country, our beloved country, because that's how you can love your country. It's not the non-serious attitude, or somebody is very rich in the university and his car is beautiful, or he is dressed beautiful or not so beautiful. That's not something which will carry on. It's the responsibility level you are going to take. So today, there will be 3 judges, as you know from the program, and they are the highly credible scientists of the country. They will be analyzing your presentations of your colleagues or your fellows, and they will score them the position number 1, number 2, number 3. And as usual, we will award. Pakistan Poultry Association is very generous. The chairman or the chief organizer of this IPEX, Dr. Asim, is very kind that they have done all possible things which industry have limited budget to do. Rather, they have done more than what. So don't run behind pencils, coupons, bags, because they are short-lived things. The big thing is that even if you have first year in your graduation program, you will still carry something that will help you in your future. So with that, I thank all organizers, particularly Vice Chancellor Professor Naseem Ahmed. And I am very lucky that I used to be his backbencher in the class. So he was in the front bench, and I'm always blessed by his kindness that he supports all of our colleagues, including myself. So thank you very much. Industry is watching you. Be careful. And and the stalwarts or the big names of the industry, as I mentioned, Miajan Muhammad Javed is here. And probably you know, one very famous, who is not with us since few years, he left us, Dr. Muhammad Sadiq. He was the real hero of our industry. So you can be hero of tomorrow. So think on those lines. Thank you.
Sir, thank you very much for the explanation and more words for the research scientists and the Pakistan Poultry Association. I'm very happy to share something with you guys. Irrespective of any news coming up, the media war, whatever, the world trust on Pakistan, Pakistan poultry industry, and the biggest companies of the world, they are visiting us, the foreigners, they are visiting us. So it really tells that how much they trust in us. They don't believe in media war. They come here, and I personally know some people from the different European countries. They are here with the families. They are here for the first time with the families to see around. So that's a very important point for the Pakistan poultry industry, and specifically for the Pakistan culture and Pakistan media. So I would like to hear one of the biggest company in the world, Avin Genetics. The whole team of Avin Genetics, they are here to participate in International Poultry Expo-IPEX. I would like to name Dr. Kamran, Technical Service Manager, Mr. Davey Tyler, Technical Service Manager, Rafael Monleon, Business Manager, Larry Blackstone, Brand Manager, Khun Nikki, Marketing Manager, Khun Ice. I'm sorry if there's some wrong pronunciation, I'm sorry for that. But Mr. Khun Ice, Marketing Assistant, Greg Hitt, Regional Technical Manager, Dominic Alfick, International Product Manager. So we would like to welcome all of you here, and I would also suggest you guys please move around and have a look and change your mind what comes up in the media. And again, thanks for trusting on the Pakistan poultry industry, which is a growing industry and will be more and more in the coming years. Now I would like to request Professor Dr. Shahid Munir, Chairman, Punjab Higher Education Commission. We are delighted to be him to be here so he can see by himself the research going on in the poultry industry. Professor Dr. Shahid Munir.
Bismillah Rahman Rahim. Honorable President of the Pakistan Poultry Association, Vice Chancellor Professor Dr. Naseem Ahmed, University of Veterinary and Animal Sciences, Honorable industrial professionals, CEOs, deans, directors, chairmen, principals, students, research scholars, Assalamu alaikum. I am very much delighted to be here as a part of this international expo and international conference on poultry science. I am delighted also to see a strong interaction and collaboration between the US and the industry. And it is a 3 days event, and I think in future, I was thinking that in universities, we can organize the chicken carnival. And I would like to discuss it with the worthy Vice Chancellor about the carnival that we can celebrate in the different universities. Ladies and gentlemen, we all know that a healthy body requires food full of nutritional components, including carbohydrates, vitamins, proteins, minerals, etc. Meat and poultry are a great source of protein. They provide a number of other nutrients like iron, zinc, vitamins, especially B12, and essential fatty acids. Poultry is classified in the white meat category. Being a low-calorie source of protein, it has added benefits for human health. Pakistan has become the 11th largest poultry producer in the world, and I think in a couple of years, it will gain its place in the top 10 producers of the poultry. According to the Pakistan Poultry Association report, the industry is the largest agro-based segment, generating employment and income for about 1.5 million people directly and indirectly. The current investment in poultry industry is more than 750 billion rupees. Poultry sector is one of the most dynamic segment of agriculture in Pakistan with a significant contribution in the GDP that is around 1.4%. Poultry sector is one of the most organized branches of the agro-based sector of Pakistan. Its growth rate is about 10 to 12%. At present, over 190 billion rupees worth of agriculture produce byproducts of agriculture are being used in the poultry feed. There are over 15,000 poultry farms spread deep into the rural areas across the country from Karachi to Peshawar. Capacity of farms range from 5,000 to 500,000 broilers. Pakistan's poultry is not less than the international standards. The poultry industry meets 50% of the total demand for meat in the country, and the rest is met by the other meat products like the mutton and the fish. Annual, we are producing 11,250 million table eggs and 1.24 million kilograms chicken meat. As per the PPA report, meat consumption per capita in Pakistan is less than the developed countries. The consumption of meat and eggs per capita is 6.2 kilograms and 56 eggs annually, whereas in developed world, the per capita meat consumption is 40 kilogram and 300 eggs annually. With the introduction of advanced technologies, more investments are coming around to cater to market needs, and unhandsome revenues in poultry industry still has a lot of potential to contribute into the economy. According to World Health Organization, a person needs 25 grams of animal protein per day, while most people in Pakistan only consume 17 grams. To meet the international standards of meat consumption, the supply and production needs to be increased and prices needs to be brought down to so that the consumers can get the required meat and egg consumption level. An increase in the production will certainly require more investment in the industry. To boost production and bring down the product rates, imports of poultry related equipment should be exempted from duties and taxes. In addition, as growers increasingly need land to establish sheds, the government should provide state land to investors at normal rates to generate investments and more products. Government should bring down the poultry feed rates to offset the price. The government should also ensure easy availability of loans to people related to the industry. Collaboration between industry and academia is a key to catalyze innovation and growth in the health and wellness of the nation. While industry often focuses on addressing solutions that are of near-term commercial value, and academia focuses on building new knowledge through research and imparting education to students, the combination can yield accelerated development of new breakthrough in this sector. Industry partnerships are instrumental in advancing research and creating a skilled workforce. Industry gain work-ready talent with specialist knowledge and practical training, and universities benefit by. People are often worried about the non-trivial values of the poultry and question about its speedy growth. Poultry industry need to address this issue. The main reason broilers are getting bigger and growing faster is genetic selection supported by the diet rich in carbohydrates and higher proteins. Besides, it involves combination of selective breeding and rearing practices. Chicken feed is also a concern of the people. People think that the feed is made up of unhealthy, non-halal ingredients. This is a serious issue. Industry also need to address this. Poultry feed is primarily a mix of corn and soya bean meal as advised by the poultry nutritionists. Corn is the primary element in most chicken diets. In fact, two-thirds of chicken feed is made up of corn. Besides this, vitamins and minerals are essential components of the chicken diet. As we are living in the era of emerging technologies, fundamental changes are taking place in health and wellness sector with personalized food, medicine, digitalization of economy and society. These will require solutions that can be deployed sustainability in Pakistan. Therefore, industry academia collaborations will be critical to innovate and commercialize these solutions in poultry sector. Pakistan Higher Education Commission will always support and encourage the veterinarian and poultry scientists for new research and innovations. UVAS and PPA are providing a platform to bring together academia, researchers, poultry farmers, poultry professionals, and industrialists. With this platform, academia and researchers will be able to share their relevant research work and learn from poultry farmers their researchable problems and current challenges with the help of process of making discoveries and then transforming them into products and services for the marketplace. So I am very much confident that this 3 days event will add a value to the researchers and the industrial professionals. And it is a matter of great satisfaction that in Pakistan now the university and industries are now collaborating for the indigenous research. With these words, I will congratulate the officer bearers of the Pakistan Poultry Association for organizing such a wonderful event. Thank you very much.
Sir, thank you very much for your kind words. Now I welcome Dr. Khalid Neem Qaida Sahab. Thank you very much for being with us. And now I would like to welcome Dr. Asim Mahmood Khan, the Chief Organizer, IPEX. Is IPEX ki jaan, IPEX ki shaan, Dr. Asim Mahmood Khan.
Bismillahirrahmanirrahim. Worthy Chairman HEC, Dr. Shahid Munir Saab, my Vice Chancellor, Dr. Naseem Saab, और मेरे colleagues और यूनिवर्सिटी से आए स्टूडेंट्स, मेहमाना निगरामी, आप सबको खुश आमदीद. It's a great pleasure and immense time for Poultry Association and IPEX 2022 that we have great personalities under this roof. And it's the first time it's happening that foreigners from abroad, they also came to join this session. Today, just we have long speeches and collaboration of different universities, associations in poultry science conference. I do hereby open this Poultry Science Conference. It is the 4th session of the Poultry Science Conference under the umbrella of Pakistan Poultry Association and International Poultry Expo. Thank you very much.
Sir, thank you very much. And I appreciate that everyone is looking for the time even. और ये मेरी ऑडियंस से भी गुजारिश है कि नाश्ता नुश्ता तो करके आए होंगे, तो थोड़ा सा नहीं करके आए जी, तो इसका मतलब है कि खाने के लिए कल से बुकें आज दोपहर के लिए, तो उसके लिए भी आपको ज़्यादा कोशिश करनी पड़ेगी. एटलीस्ट यू मस्ट पुट सम एफर्ट ऑन द क्लैपिंग इवन. थैंक यू. एक दफ़ा करके बताइए ज़रा. थैंक यू वेरी मच. इट मीन्स एवरीबॉडी इज़ लिसनिंग. आपकी क्लैपिंग जो है इट असिस्ट द स्पीकर्स बेसिकली, स्पेसिफिकली द रिसर्च स्कॉलर्स आर कमिंग ऑन स्टेज. आप उनकी बात सुने, अप्रिशिएट करें, सो इट वुड एनकरेज दैम. एंड आई ऑल्सो वेलकम डॉक्टर फयाज कमर साहब हु इज़ विद अस नाउ. And now I would like to request to come on stage as Professor Dr. Kashif Salimi Saab. Please come on stage and conduct this session now. Thank you, sir.
Bismillahirrahmanirrahim. Before start of session, I will request speakers of first session to please come in the second row, ताकि हम आसानी से उनको बुला सकें. जो स्पीकर्स हैं फर्स्ट सेशन के, अगर ऑलरेडी सेकंड रो में हैं तो वेल, अदरवाइज़ वो सेकंड रो में आ जाएं. तो हमारे गेस्ट बैठे इनसे सेकंड वाली रो में, ताकि हम इजीली उनको बुलाएं, ताकि टाइम वेस्ट ना हो. I welcome all of you in the formal session of Poultry Science Conference 2022. Before start, I will request Professor Dr. Anas Sarwar Qureshi, Pro Vice Chancellor, University of Agriculture, Faisalabad, who will be the chair of this session. Sir, please come on the stage. Professor Anas Sarwar Qureshi has a long experience of teaching and research. Now I will request Professor Dr. Masood Akhtar. He is having an experience of more than 30 years research and teaching. Dean Faculty of Veterinary Science, Bahauddin Dikri University, Multan. He will be the co-chair of this session. I will also request Professor Dr. Sarzameen Khan. He is Dean Faculty of Veterinary and Animal Sciences, Agriculture, University, Peshawar. He will also be co-chair. I will request Professor Dr. Mazhar Ayaz. He is our guest of honor, Professor of Pathology in Parasitology in Chulistan Veterinary University, Bahawalpur. We will have 10 minutes for presentation. So this is my request to the speakers, please stick to time. Sir, with your permission, we are going to start with the session. Our first speaker is Mr. Hafiz Muhammad Salman Khalid from University of Agriculture, Faisalabad. Okay. Our first speaker is our keynote, Professor Dr. Ahmadeen Anjum. Professor Dr. Ahmadeen Anjum is one of the leading poultry pathologists globally, and he remained a long chairman Department of Pathology, University of Agriculture, Faisalabad. And this is my honor that he is my teacher, Professor Dr. Ahmadeen Anjum. He is one of the leading person, and he is a like डॉक्टर हनीफ नज़ीर साहब ने जैसे बताया था मियाँ जान मोहम्मद जावेद साहब के बारे में कि वो पोल्ट्री इंडस्ट्री के बिग जाइंट हैं, तो डॉक्टर एडी अंजम पोल्ट्री पैथोलॉजी और पोल्ट्री डिजीज़ में दुनिया में एक बड़ा नाम है. प्रोफेसर डॉक्टर अहमदीन अंजम, प्लीज़. चेक करें सर.
Hello.
Assalamu alaikum. It's an extreme honor for me to deliver the keynote lecture on poultry health to this poultry science conference. I am really grateful to the organizers who provided me this opportunity to share my experience with the leaders in the industry, leaders in the academia, my colleagues and my students. This occasion will foster cross-fertilization of ideas among farmers, food scientists, poultry veterinarians, pathologists, microbiologists, and immunologists. For me, this session is in fact addresses the societal demands of the society on safe poultry meat and eggs, and I really appreciate efforts of the PPA who supported PPA and the PPA who worked for the poultry industry and society. First of all, because this session is on the health, let us see what is health. The one which is a long accepted definition of the health is it's a state of norm. This is what FAO said in 1946, but then they had to explain it in 48 that it's a complete physical, mental, and social well-being. But again, this does not apply to chicken. So what is health in the chicken? And again, it's my own explanation. For the chicken, if the outcome is profitability, this is health, and this involves the least cost production. If you look at background of the chicken, look in the first instance, this chicken was, sorry, there is no marker, but we selected the chicken from wild jungle fowl and we went for the intensification. So the first change was selection and intensification. In the wild, there was a group of 12 chickens while we put 35,000 in a single house. And the second change, what we did, they were laying only 4 to 6 eggs twice a year. And now we have forced them to produce more than 320 eggs. And same we did with the broiler. The message here is that we changed their behavior, we changed their anatomy, and we changed their physiology. With this, we pressurized their health, and what came up is the production diseases. And this production diseases, when I tried to look, other than my own experience, I looked what my colleagues are doing and what are problems of the industry, but unfortunately, I had to look into the published papers. I couldn't find anything on the prevalence of the diseases. I looked into activity of the veterinary institutions as well as universities, research institutions, but there was not much. I couldn't find what are the problems of Pakistan Poultry Association. So the problem is that we don't know our problems. And to know our problems, I suggest a model of a poor country, Nigeria. What they did, just they developed a small app on the mobile, and they gave access to the farmers and to various institutions, and they generated the data which can be categorized as, and if we do that, this then with the passage of time, there are 3 types of technology which even farmer can use. So the very small technologies like this again from Nigeria, that the farmer on the left picture is just photographing the fecal droppings, and they fed it to the computer, and then later on they developed a database, and even the farmer can classify at least 3 of the diseases. Again, sorry, Dr. Sir, the marker is not working. So there are 3 technologies now on the way which we can add later. First, we need to develop a simple app, and then we need to equip it with the disease diagnosis simple techniques which a farmer and field veterinarians can use. If we look at the available data, there are 5 diseases which are in the viral categories: Newcastle, Gumboro, adenoviruses, and H9. They are circulating widely in the field. And in the bacterial diseases, again, Mycoplasma, E. coli, Coryza, Salmonella, and cholera. They are the leaders. And in a miscellaneous component, mycotoxin and cocci, they are the major players. But I have categorized this into 3 groups. If, for the health purpose, if the industry can take care of 3 things: first, the respiratory system, health of the respiratory system, health of the intestines, and immunosuppressive diseases. So this, we need to understand that these are the 3 blocks which are causing health problems. In the respiratory tract component, if we see there is damage to the respiratory epithelium with the environments, and then first entry is of the viruses, that goes with the E. coli, and also there is E. coli. What we see is the end on the downside, and that is respiratory disease complex. That complex, if what we see, we treat that. But that does not give us any response because the other components are ignored. So the message is that we will have to look for the prevention as a block, not as a single entity, what we see at the end. So this is the second group. And again, this is very alarming. In Karachi, this is a report that H7 is also circulating in Karachi flocks, poultry flocks, alone in combination with Newcastle disease. So this is all outbreaks which are pathogenic, thereby H7. But all H7 is not highly pathogenic. So if this is circulating in the Karachi, then anytime it can come out, and this can cause problem. This is another interesting situation for the IB in the respiratory group. We know it causes respiratory disease. We know it damages kidney, and we know that it causes false layer syndrome. But we don't know, most of us, that in the males, it damages testicles, and somebody has seen right on the right side of the picture, the unaffected males, and compared their testosterone level with the infected males, and any male which has less than 1 milligram, less than 1 nanogram per mil of testosterone, that is infertile. So we can use this information while artificial insemination, when the males are short, we can further test and use this tool. Any male with less than 1 nanogram per mil will be infertile. Then there are enteric complex, and in this, there are 300 possible potential interactions. In these interactions, we have 2 main players, coccidiosis and necrotic enteritis. And here we can see that coccidiosis that damages the intestine, produces serum rich in proteins, and those proteins invite the Clostridia. So when we are looking for the enteritis and enteric disease complex, if we don't address the two together, if we only treat coccidia, there will be no result. Only treat Clostridia, there will be no result unless we address both of them. In the immunosuppressive, we have four candidates circulating: adenoviruses, Gumboro, infectious anemia, and mycotoxins. We know this component of the this hydropericardium and IBH before, but what I have experienced now, there is a recent invasion. This is adenovirus gizzard erosions, and this is very interesting virus. Look, if we start from the chicken, adults, the virus is circulating present in their oviducts. It enters the eggs, and from the eggs, in the neonated chicks in the embryos, it propagates. From the embryos, it goes to the day old. From the day old, it is excreted right from the day old. Even in the hatchery boxes, it is excreted in the trays, and then this is how it is circulating, and I call it as a hidden enemy. And this is but this is another important feature. Look, I injected some of the liver homogenate, and look at the intestines of the 11 of 17 had this hemorrhagic Zika. This is a new, I have not reported it, I have not seen it. This was FAD, fowl adenovirus serotype 2. And this is something to be investigated. Otherwise, what is creeping, how is it changing, we don't know. So Gumboro, you know, chicken anemia, you know. So the message is that there are co-infections. Even in immunosuppression, we need to take care of this. So this is of zoonotic importance. We have no contribution for this, and there is no awareness, and we are not have no programs to give the safe to the. Similarly, for the rural poultry and for the pet birds, this is also a threat to the poultry health, and we need to address that or leave it as a danger. So who will do all this? I think the owners, the vets, the government, and the association, all these leaders, these need to work together to further facilitate and improve health of the poultry flocks. Thanks.
Thank you, sir. Thank you very much. We have a little change in the program. I will request Professor Dr. Naseem Ahmed, Vice Chancellor, University of Agriculture, Faisalabad, to come on the stage to present shield to our chief guest, Professor Dr. Shahid Munir, Chairman, Punjab Higher Education Commission. I will also request Dr. Rana Sajad Arshad, Chairman, Pakistan Poultry Association, and chairs and co-chairs of this session to accompany Professor Naseem Ahmed. Please. Actually, Dr. Shahid Munir sahab ki aur assignments hain. He has to leave. Therefore, we have a little change in the program. Thank you very much, sir. Thank you. Thank you, sir. Okay, we will continue with our session. And our next speaker, Hafiz Muhammad Salman from Department of Pathology, University of Agriculture, Faisalabad. He will speak on study of co-infection of chicken infectious anemia virus with fowl adenovirus in commercial poultry. Salman, please. Salman, you have 10 minutes. आपने यही माइक यूज करना है, थोड़ा सा पीछे हट के ये नहीं करना है, इससे वो साउंड का प्रॉब्लम बनता है. 10 मिनट है, स्ट्रिक्टली. ठीक है.
अस्सलाम वालेकुम, बिस्मिल्लाहिर रहमानिर रहीम.
एरो से मूव करना आपने, ठीक है.
I am Hafiz Muhammad Salman Khalid. Currently, I am doing PhD and working as research associate under the supervision of Dr. Muhammad Kashif Salimi from Department of Pathology, University of Agriculture, Faisalabad. The topic that I am going to present was my MPhil research, that was study of co-infection of chicken in anemia virus with fowl adenovirus in commercial poultry. Chicken anemia virus belongs to genus Gyrovirus from the Anneloviridae family, first identified in Japan from contaminated Marek's disease vaccine in 1979. Fowl adenovirus belongs to family Adenoviridae and genus Aviadenovirus, that is double-stranded DNA virus. Aviadenoviruses are classified into 5 species, fowl adenovirus A to fowl adenovirus E, which are further divided into 12 serotypes. Fowl adenovirus is the causative agent of different disease conditions in poultry like inclusion body hepatitis, hepatitis hydropericardium syndrome, and hydropericardium syndrome associated with hemorrhages and necrosis of the liver. Chicken infectious anemia is an emerging disease of the poultry, causing considerable economic losses to poultry industry, and its co-infection with fowl adenovirus usually produce more pronounced and lethal infection in birds. To explore the epidemiological status of CAV, FADV, and their co-infection, sampling was done from commercial poultry flocks across Faisalabad division. Samples including spleen, liver, thymus, bursa were collected aseptically and further processed for DNA extraction. The extracted DNA was then quantified by using nanodrop and subjected to amplification through polymerase chain reaction. Diagnosis of the FADV based on the hexone gene and for CAVVP2 gene was targeted. And the analyzed amplified product were then visualized in a 1.5% agarose gel stained with ethidium bromide. And for histopathology, tissues including liver, spleen, thymus, bursa, and lungs were stored in 10% neutral buffered formalin for proper fixation of the tissue. After that, the sections were stained with the hematoxylin and eosin technique and then evaluated for histopathological alterations. Here is the first image. We can see that these samples were positive for fowl adenovirus at 486 base pair position. In second gel, these 4 samples were also tested for chicken anemia virus. We can see that the first one is the ladder and the second lane in the control positive and third one is the control negative. Then these next 4 samples were positive for fowl adenovirus at 486 base pair position. And these samples were also run for chicken anemia virus and these samples were also positive for chicken anemia virus at 186 base pair position. A total 640 samples were collected across the Faisalabad division. From Faisalabad, 570 samples were collected, in which 13.2 samples were positive for CAV and 7% samples were positive for fowl adenovirus and 2.6% co-infection was observed. And from TT Singh, Topatek Singh, 14.3% co-infection percentage was observed. Here is the seasonal wise co-infection percentage, in which we can see that maximum co-infection was observed during July to September. While talking about the age-wise co-infection percentage, maximum co-infection percentage was observed in birds having age 11 to 20 days. And while talking about the mortality trend, the maximum number of the birds with CAV infection observed mortality 150 to 200 per day. While in case of co-infection, this mortality was increased up to 300 per day. And while talking about the geographical co-infection percentage, maximum co-infection percentage was observed in Toba Tek Singh district. And here is the, you can see the histopathological figures, in which first figure you can see the bursal parenchyma in CAV positive birds showing lymphocytic depletion in both cortex and medulla. In second image, hepatic parenchyma of CAV positive birds showing congestion and individual cell necrosis. In third micrograph, we can see the liver of the CAV positive birds showing cellular infiltration, vacuolar degeneration, cell swelling, and individual cell necrosis. In fourth micrograph, the bursa of the CAV positive birds showing mild lymphocytic depletion both in cortex and medullary region. In the fifth micrograph, we can see the thymus of the CAV positive birds showing lymphocytic depletion, congestion, and no differentiation and demarcation between cortex and medulla. In the last micrograph, we can see the spleen with indicating lymphocytic depletion. The granular material with vacuolar spaces are present. The study concluded that both chicken infectious anemia and fowl adenoviruses are present in commercial poultry flocks. The co-infection of fowl adenovirus with chicken anemia virus leads to high mortality and severe immunosuppression, as both these agents are known as immunosuppressive viruses. As both diseases are vertically transmitted, so therefore effective vaccination of predator flocks to prevent the vertical transmission of disease in the progeny is very important. So thank you very much.
Thank you very much, Salman. You are well before time. Now our next speaker, Dr. Shahan Azim. He is Assistant Professor in Department of Epidemiology and Public Health, University of Veterinary and Animal Sciences. And Shahan will speak on environmental sampling for avian influenza virus detection in commercial layer facilities. Shahan, please.
Thank you very much, Dr. Kashif Salimi. I appreciate the committee that selected my abstract and allowed me to share my findings about the environmental sampling for avian influenza virus. So the background to this study was that we know that avian influenza virus can cause a devastating disease in poultry. And we know that especially in the endemic countries such as in Pakistan, we have this H9N2 that is causing havoc in the poultry industry because it's low pathogenic, hard to detect, keeps spreading the disease, and we don't know, and we have the production losses and economic losses. So the solution to this that came to our mind was that we can do some environmental sampling, and in this study we did 2 experimental studies, and there was 1 field study. So what we did was, where we find out that if we can do the environmental sampling, we don't have to do the sampling from the birds directly, no pain to the birds. We can just select some samples from the environment, we do testing on them, we detect avian influenza, and we can declare if the farm or the premises is positive for avian influenza or it is not. And we have done this, and we found out that environmental sampling is useful, it's effective, it's accurate, and it is convenient, and as well as it meets the animal welfare standards. So in the first experiment, what we did was we selected some of the environmental sampling matrices. We collected the samples with Swiffer pads, drag swabs, as well as water. We selected samples from the commercial facilities, and then we spiked them with the avian influenza virus, a low pathogenic avian influenza virus, and then we incubated these samples at 4 different temperatures for 4 different times, and at the end of the experiment, our goal was whether can we detect an influenza virus in them. Is there a difference between the sample matrices in detection of the avian influenza virus? Is there an effect of the sample matrices on avian influenza virus testing? So that was our first experiment. In the second experiment, what we did was we evaluated if the sampling materials or collection devices have any effect on the avian influenza virus detection. So we chose the boot swab, we chose the Swiffer and drag, as already mentioned, and we spiked those with the virus, and then we spread the virus on the floor, collected with these collection devices, and try to determine which of these is a better collection device, or is there any difference or not. And in the third or the last study, what we did was, and we did this in two different ways. So one was the limit of detection for the first study, and the second one was the half-life. I will explain more what is the limit of detection, what is the half-life when I talk about the results. So in the field study, we collected samples from the field, and we selected different locations, for example, egg belts, cages, manure pits, manure belts. We selected these, and we wanted to make sure or understand, ascertain which of these is a better sampling location. Is there any difference, or there is no difference? And then the other thing we saw was that is there any virus-wise, virus CT-wise, virus titer-wise, is there any difference between any of the sampling locations? So these were our three main experiments, and let's talk about the results now. So in the first study, what we found out was that, and I have to go back a few slides for the results. So in the first study, we found out that the limit of detection increases with the temperature. So we know avian influenza virus is a temperature-sensitive virus. If there is a higher temperature, if the virus is stored at a higher temperature, the LOD is going to be increased. And therefore, we have to keep the samples, environmental samples, when we collect them, we have to keep them chilled before we transport them to the lab, even during collection and transporting them to the lab, keep them chilled. Another thing that we observed was that environmental sampling also told us that the samples are good enough. We compared it with the DMEM, which is an ideal storage medium. We compared it with that, and we found out that environmental samples, they preserved the virus. The virus did perfect in those samples. In the second study, we saw that what is the half-life? Half-life is the time which virus takes to decrease in number to 1/2. So in this study, what we found out that the half-life also is short at the cold temperature, and it's longer at the higher temperature. So that is also, the temperature effect on the virus is also seen in this. And then in the second, what we found out that the collection devices, they almost did the same. They were similar. There were no statistical differences between the collection devices. We can choose any of the device as per our convenience, because they were equally good in collecting environmental samples. However, economics is the matter of concern. We can choose the device which is least expensive so that we can save money. And in the final and the field study, what we found out that certain locations, for example, manure belts, egg belts, cages, house floors, and manure pits, they were more positive for the virus, meaning they should be targeted if we have to collect environmental samples. So, wrapping up, finally what we found out that environmental sampling is convenient, it's accurate, we can use it for surveillance of the poultry facilities, because if we do the surveillance in a timely manner, we can control the spread of the disease. Especially this could be useful in our circumstances, because once the flock is out and there is a downtime, before restocking, we do the environmental sampling to make sure that the house is clean, so that we can repopulate the flock with the surety that we are not restocking in an infected premises or infected house. So what we found out that LOD and half-life, the data, they both told us that we need to maintain a cold chain before we can bring the samples to testing for the laboratory, so that we can have a best results. Thank you very much for your attention. This is the paper that came out in the proceedings. If you need more details in the proceedings, page number 2 or abstract 2 is where it is. I asked one of our friends if you need some handouts, so Sarfraz will pass it on to you. Please raise your hand. Thank you very much once again.
Thank you, Dr. Shahan. I have two housekeeping announcements. One is that if anybody want to ask any question from our speakers, there is a prescribed pro forma. हाल में जो हमारे मैनेजमेंट के लोग हैं उनके पास क्वेश्चन है जिस पे आपने स्पेसिफिकली क्वेश्चन पूछना है. ऑरेंज जिन्होंने ऑरेंज शर्ट्स पहनी हुई उनके पास परफॉर्मा है जिससे आप क्वेश्चन पूछेंगे और क्वेश्चन आंसर सेशन में हम सिर्फ इन्हीं को एड्रेस करेंगे. सेकेंडली, इसके हॉल के बैक साइड पे वाशरूम्स हैं, टॉयलेट्स, अगर किसी ने यूज़ करना हो तो बैक साइड पे हैं. वी विल कंटिन्यू विद दिस सेशन. आवर नेक्स्ट स्पीकर इज डॉक्टर मोहम्मद सईद from University of Agriculture, Peshawar. Dr. Saeed will speak on pathogenesis, isolation, and molecular characterization of Mycoplasma gallisepticum in naturally infected broiler chicks. Dr. Saeed, please. You have 10 minutes.
Bismillahirrahmanirrahim. My name is Dr. Mohammad Saeed. I am from the College of Veterinary Sciences, the University of Agriculture, Peshawar. The topic of my presentation is pathogenesis, isolation, and molecular characterization of Mycoplasma gallisepticum in naturally infected broiler chicks. Introduction: poultry sector is one of the most organized sector in Pakistan. This sector generates employment for about 1.5 million people. Poultry meat contribute 26.8% of the total meat production. The poultry industry in Pakistan is facing serious challenges in form of poor management, harsh climatic conditions, fatal disease outbreak. Among various diseases, Mycoplasma is the most prevalent and challenging to the poultry industry. The etiology of the mycoplasmosis is Mycoplasma gallisepticum, Mycoplasma synoviae, and Mycoplasma meleagris. But among the Mycoplasma gallisepticum is the major cause of chronic respiratory disease and economically causes more losses to poultry industry. Their classification is kingdom Bacteria, phylum Pterygoids, class Mollicutes, order Mycoplasmatales, family Mycoplasma mutaceae, genus is Mycoplasma. Mycoplasma gallisepticum considered to be the most pathogenic of all Mycoplasma pathogens. Their signs and symptoms are there will be rales, coughing, sneezing, nasal discharge, dyspnea, and lacrimation. There will be there is a decreased weight gain and egg production, and also decreased feed efficiency. The gross lesions, when we see to the post-mortem examination, there will be mild sinusitis, tracheitis, air saculitis, thickening and turbidity of the air sacs, accumulation of exudate in the lungs, pericarditis, perihepatitis may be seen, swollen and inflamed eyelids, ocular and nasal discharge. The microscopic lesion of chronic respiratory.
Excuse me, मेरी तमाम स्टूडेंट्स से गुजारिश है, काइंडली आप अगर इफ़ यू कुड लाइक टू हैव सीट्स प्लीज़. ये स्पीकर के लिए बहुत ज़्यादा डिस्टर्बेंस है इफ़ यू कीप मूविंग अप एंड डाउन. और एक अनाउंसमेंट भी है कि अगर रेस्ट रूम या वॉशरूम्स जो हैं वो देना डाउन स्टेयर वो ऊपर ही हैं, इसलिए आप नीचे हॉल के जो ना वो डोर की तरफ मत आएँ. बहुत शुक्रिया आपका. ये जो स्टूडेंट्स जो अपर साइड पे स्टेयर्स में हैं, काइंडली कुड यू प्लीज़ हैव द सीट्स प्लीज़. और अगर ऊपर चेयर्स नहीं हैं तो काइंडली से नीचे तशरीफ़ ले आइए. थैंक्स फॉर द कोऑपरेशन.
Microscopic lesions, necrosed and swollen tracheal epithelium. There will be leukocytic infiltration in mucosa of trachea and sloughing of the cilia in mucosa, and there will be hemorrhages of variable degree. Lungs shows congestion, hemorrhages, and focal necrosis and leukocytic infiltration. Hemorrhages, degeneration, and necrosis of hepatocyte, degeneration and of cardiac muscle and pericarditis. Diagnosis, how you will diagnose the CRD, chronic respiratory disease. First, isolation of mycoplasma is difficult because of variation in the clinical sign and symptom. And the second most important about the mycoplasma, it is a fastidious bacteria for growing, and their growth is very slow and need a special media for growing. There are so many different tests are recognized, but PCR is the most accurate and sensitive molecular technique for the confirmatory and diagnosis purpose. The objective of our study is isolation of Mycoplasma gallisepticum by using special media, confirmation of Mycoplasma species through PCR. Our third objective was study the pathogenesis of the disease by recording histopathological changes in naturally infected birds. Our study the study was conducted in Peshawar, KPK. Total 250 samples were collected from the naturally infected birds. Our experimental design was that from the live birds, nasal swab and tracheal swab were collected, and from dead bird, the lungs, trachea, and liver were collected. From live birds, we cultured the nasal and tracheal swab in a special media in PPLO broth, and after this, we place the culture in the incubator, and after that we extract the DNA and confirm through PCR. And from the dead birds, we take the lungs, liver, and trachea and preserve in 10% neutral buffer formalin and perform the histopathology. A total of 50 samples for histopathology and culture were collected from the birds exhibiting the signs of the respiratory disease. Nasal and tracheal samples were collected through sterile nasal swab for bacterial isolation. A sample from the tissue, lungs, liver, trachea were processed for histopathological study in the Mycoplasma Reference Lab, the Department of Animal Health, the University of Agriculture, Peshawar, for onward processing. For isolation, we use the PPLO broth and PPLO agar media, and we confirm through these by the color change and turbidity and whirling movement in culture, colonic characteristics, and last we confirm through the PCR. This is the composition of the PPLO medium. There are two portions of the PPLO medium. The one is the autoclavable portion, the second one is the filtrable portion. In autoclavable portion, we use the PPLO broth, deionized water, and if we go for the agar preparation, then we add the PPLO agar. In case of filtrable portion, horse serum, first we inactivate at 56 degrees centigrade for 30 minutes. Yeast extract, glucose solution, sodium pyruvate, and antifungal and antibacterial penicillin were used. And the pH were adjusted to 7.8. Inoculation of a sample in PPLO broth media, and we place the culture in CO2 incubator at 37 degrees centigrade for 5% CO2. Change in color from the red to yellow was regarded as positive sample by only cultivation. Positive sample was streaked on solid agar medium with the help of sterilized loop. Agar plate were examined daily up to 6 days after the culturing. So Mycoplasma shows a typical fried egg-like colonies. The DNA was extracted through the kit from the culture, and the specific primers were used for Mycoplasma gallisepticum having 185 product size. A total of 25 microliter PCR reaction was prepared. The composition is reverse and forward primer 1.75 microliter, master mix 10 microliter were used, nuclease free water 8 microliter, and DNA was template 3.5. PCR condition was first initial denaturation was 95 degrees centigrade for 3 minutes followed by 34 cycles, and we optimized the annealing temperature at 54 degrees centigrade for 30 minutes and extension for 72 minutes. We prepare the 1% gel for the PCR, add in TAE buffer, and add 2 microliter of ethidium bromide. For histopathology, we take the samples and preserve in 10% neutral buffer formalin and followed the standard protocol as adopted by Bancroft and Gamble. Data was simply analyzed by simple percentage results. Through culture, 120 sample were positive and 130 were negative, and when these positive samples were subjected to PCR, it gives 64% samples shows positive result on PCR. This is the picture of the culture. The red one shows the control one, while the yellow shows the change in the pH. When there is a change in the pH, there will be color change, and this shows the turbidity, and also when by shaking, it gives you a whirling movement. This is the typical mycoplasma fried eggs colonies. We picked these colonies and cultured and confirmed through the PCR. This is the PCR result. We use the 1 kb ladder having the first band was 250 base pair, but our target was 185 product size. This is the histopathological findings. The trachea shows the sloughing of epithelia, and also there was some disruption in the cilia of the trachea, and also leukocytic infiltration. This is the histopathology of lungs that shows the arrow shows the mucus plugs, the emphysema, and this is the second one. Is also the lung slide shows the emphysema in lungs. The liver, this is the our first finding was that the liver shows some changes through when we see the microscope. There was the leukocytic infiltration, necrosis of hepatocytes, and the central vein was blood engorged. So our study of, the conclusion of our study was that MG Mycoplasma galisepticum was first time confirmed through PCR in KPK. Along with respiratory tract, the lesion were also observed in liver, which indicate that the multisystemic involvement of the disease. 53% confirmation of disease through PCR indicate the widespread of nature of disease in District Peshawar. We recommend that the study is needed to be conducted to sort out all other species of mycoplasmosis in birds for control of this disease. DNA sequence is most important. We need DNA sequencing for further prevalent in KPK. Further study is recommended to be carried out on vaccine production against CRD. Standing operating procedure may be introduced at poultry farms, markets, and selling points. The last one is our recommendation that PCR can be successfully used as a reliable diagnostic tool for screening of flocks for disease control. Thank you.
Thank you, Saeed. Our next speaker is Ms. Maria Jamil from University of Agriculture, Faisalabad. Maria will speak on immunopathological effects of avian adenovirus infections in broilers fed with aflatoxin B1 contaminated feed.
Bismillahirrahmanirrahim. Assalamu alaikum. I am Maria Jamil, PhD student in Department of Pathology, and I am coming from University of Agriculture, Faisalabad. Today I present my MPhil research work that is about the immunopathological effect of avian adenovirus infection in broiler bird fed with aflatoxin B1 contaminated feed. First of all, introduction. Contamination of poultry feed with fungi and their metabolite is well known as mycotoxin, and it is the global issue. Mycotoxin, these are secondary fungal metabolites produced by various toxigenic fungi, especially belonging to the Aspergillus parasiticus and the Fusarium. And according to the World Health Organization, 25% of world crop, these are contaminated every year due to the mycotoxin. Mycotoxin, these are well known as invisible thief, silent killer, unavoidable contaminant, and the natural toxicant. And there are certain mycotoxins that gain the critical importance regarding to the health status of human, animal, and the bird, some of which are aflatoxin, ochratoxin, zearalenone, and fumonisin. And among all of these, my main focus is on the aflatoxin. Aflatoxin, it is most common mycotoxin produced by the various toxigenic fungi. And aflatoxin, these are produced by the Aspergillus flavus and Aspergillus parasiticus. And these are further categorized into 6 subfamily on the basis of the ultraviolet light. When passed through it, it produces the color. And among all of these, aflatoxin B1 is the most common mycotoxin, and according to the World Health Organization, it is categorized into the group 1 carcinogen. According to the Biomin Mycotoxin Survey 2021, total risk of the mycotoxin, that is 95%, and the prevalence of the aflatoxin, that is 92%. Now about the avian adenovirus infection. Avian adenovirus infection was firstly isolated from the clinical disease in quail in 1950, and in Pakistan, firstly avian adenovirus infection was identified in 1987 at the Angora goat Sin perfins. Avian adenovirus is double-stranded DNA virus belonging to the Adenoviridae family. And Adenoviridae family having the 5 genera, but my main focus is on the AV adenovirus, genus AV adenovirus, that is further categorized into the 5 type A to E, and on the basis of the vast range of virulence and the pathogenicity, and having the 12 serotype, but my main focus on the type C, that having the serotype 4. Now about the economic losses, both about the mycotoxin and avian adenovirus infection. According to the World Health Organization, 1/4 of the crops, these are contaminated every year due to the mycotoxin, and annual cost of the mycotoxin, that is around about $5 billion US. And it has been estimated that the economic losses due to the mycotoxin, that aflatoxin B1 that has been exceeded to the 143 billion, and each milligram of the aflatoxin B1 would lead to decrease the growth performance of the bird, that is around about 5%. And it has been estimated that the adenovirus infection, it lead to the morbidity 100% and mortality 30%, and immunocompromised bird, it lead to the mortality 80%, and the growth losses in the enteric flock. Now about the main purpose of this research work was to investigate the immunological effect of the aflatoxin B1 in the bird co-infected with avian adenovirus infection, and the pathological effect of avian adenovirus infection in the bird intoxicated with aflatoxin B1 contaminated feed. This is the layout of my experimental design. For this experiment, I was taken the total 120-day-old bird. After 2 to 3 days of acclimatization, these birds were divided into 6 groups. Each group having the 20 birds. Group A was control group. Group B and C was given the aflatoxin B1 in dose-related manner, and the group D was given the avian adenovirus infection, and group E and F was given the co-infection. For the preparation of the experimental feed, fungal contaminated feed was collected from different forms and then analyzed by the HPLC for aflatoxin B1. And the mycorrhizal contaminated feed were prepared by diluting this feed in the basal diet. And the avian adenovirus infection was inoculated in the bird at the 15th day of the experiment at the dose rate of 0.5 ml per kg body weight in the breast muscle. And the parameters that I was studied during this experiment were included the production parameters such as clinical sign, feed intake, body weight, absolute and relative organ weight, and the immunological parameters such as sheep red blood cell, carbon clearance assay, phytohemagglutinin P, and HI antibody titer against the Newcastle disease, and the gross and the microscopic pathology of the immune organ. Now about the result, clinically birds were appear normal in group A and B, while the birds in group C and F were dull, depressed, ruffled feather, pale comb and water, less attraction toward the feed and more attraction toward the water. Highest mortality was observed in co-infected group, that is F, that is around about 60%, and in separately treated group C and D, mortality was observed 20%. Weekly body weight gain was also observed, and the marked reduction was observed in co-infected group and also in the separately treated group in dose-related manner from the week 4 to 5th. And also feed intake was observed. Feed intake was markedly declined in co-infected group, also in the separately treated group in dose-related manner from the week 3 to the 5th. And the macrophage capability was also determined by the carbon clearance assay. And it was observed that the co-infected group and aflatoxin treated group take more time to clear the carbon particle after 3 and 15 minutes of injecting the black Indian ink in the wing vein. And the lymphoproliferative response to phytohemagglutinin P was also observed, and the thickness was markedly declined in the co-infected group, also in the separately treated group in dose-related manner after the 24 and 48 hours of injecting the PHAP in the interdigital space. This is the graphical presentation of the antibody response against the sheep red blood cell. Antibody titer was markedly declined in the co-infected group, also in the separately treated group in dose-related manner, both at the 7 and 14th day of injecting the primary injection, and the antibody titer was also markedly declined at the 7th and 14th day of injecting the booster injection. Now about the pathological lesion. Grossly, it was observed that liver become pale, anemic, enlarged, and also the hemorrhages was also observed in the liver. And in the pericardial sac, you can see the straw color fluid. And now about the microscopic examination. Group A was the control group and showing the normal photomicrograph of the bursa, and the aflatoxin treatment group, that is B and C, in which red arrows show the disruptive epithelium and the black arrows show the thickest interfollicular connective tissue, and arrowheads show the lymphocytic depletion. Group D, that is infected with avian adenovirus infection, in which black arrows show the thick interfollicular connective tissue and the arrowheads show the lymphocytic depletion. And in the co-infected group, you can see red arrows show the thickest interfollicular connective tissue and the blue arrows show the lymphocytic depletion. Here, group A show the normal photomicrograph of the thymus, and group C, that is the aflatoxin treatment group, in which blue arrows show the congestion in the medullary region of the thymus, and group D show the red arrows show the congestion and the blue arrows show the lymphocytic depletion, and in co-infected group, blue arrows show the congestion. Now about the conclusion. The result indicated that the bird fed with aflatoxin B1 alone or in combination with avian adenovirus infection in dose-related manner produced the severe pathological lesion and reduced the immunological response. Thank you so much.
Thank you, Maria. Now our next speaker is Mr. Ahad Fiaz from University of Agriculture, Faisalabad, and Ahad is going to present on molecular characterization of infectious bronchitis virus, its variants, strain leading to pathogenesis of host immune response in the white leghorns. Ahad, please.
Bismillahirrahmanirrahim. Thank you once more for the introduction, Dr. Muhammad Kashif. I would like to reintroduce myself. My name is Ahad Fiaz. I recently completed my PhD from Department of Pathology, University of Agriculture, Faisalabad, under the supervision of Dr. Muhammad Kashif Salimi. Now, first of all, I will tell you about the importance of poultry. Now, I will not go into the figures or the values. I will tell you about the importance of poultry, the grassroots level to the end consumers. What do we hear when we listen to the word poultry? Usually meat and eggs. What does meat provide us? Back about 15 years ago, meat was considered delicacy, only served to the VIP guest or when someone was sick, but not now. The scenario has totally changed. Meat is served on daily basis to everyone, and everyone is consuming poultry meat daily. If we talk about the eggs, the PP has recommended 2 eggs per day as it is an economical and cheap source of protein and fulfill our nutritional requirements. So that's a point in case of eggs. And if we talk not but least, if we talk about the vendors or the industry linkages, you can say the poultry is now recognized by the industry as different vendors have named their business for the name of poultry, like the Kentucky Fried Chicken, the AFC. In my hometown, Faisalabad, 8 to 10 small vendors or the multinational business have named their business according to the poultry names, chicken industry. But you know, anyone who is doing good, there are enemies or foes. And in case of this disease, or in chicken poultry, one of our enemies is the infectious bronchitis virus. And this infectious bronchitis virus had many accomplices, like many partners that are helping the spread of the disease. It is a horizontal disease, so it usually spreads by the fomites, wild migratory birds that are usually, this is the season right now, the birds are coming, and the mismanagement, the vaccination failures, this all sum up in causing the infectious bronchitis virus. So let's start with the presentation, that is molecular characterization of infectious bronchitis virus variant. This is just a part of my thesis. I only took 2 or 3 parts from my thesis, which I'm going to present here. So if we talk about the global prevalence, it is prevalent worldwide where the intensive poultry farming, meaning where there is heavy poultry industry, infectious bronchitis virus will spread. And the incidence of approaching 100%. Now vaccination is only partially successful. Why? It is a coronavirus. It belongs to the genus Gamma coronavirus. As you have already seen in the history, in past 2 or 3 years, we have infected with the coronavirus, and that's the alphas, belongs from the alpha, but our poultry, our chicken infectious bronchitis virus belongs from the Gamma coronavirus. So we are continuously being immunized by different vaccines, and that's the same case scenario seen in the poultry, that the antigenic variation occurs and the vaccination is not properly successful. About 50 different IBP genotypes have been identified. It belongs to the RNA virus. It's round to pleomorphic in shape, and it's covered with an envelope 120 nanometer in diameter, usually surface covered with the surface projection called spikes. The target tissue for the infectious bronchitis is the respiratory tract, usually resulting in the nasal discharge, coughing, rales, and gasping. The infection causes pathological changes in the kidney, oviduct, and gonads, also resulting in nephritis and reduced egg production. And if we open the bird, we do the postmortem, we can see there are water-filled cysts that usually cover up the abdomen, take all the space, and the bird is showing penguin-like appearance. Like the penguin walks, the layer will also walk like that. Now for the sample collection, we collected about 2720 samples from Faisalabad division, stored in the RNA letter, then later processed for the RNA extraction, and we used 3 different primers. One primer on the above right corner is the S1 partial gene, while the below one are 2 are the full S1 genes. After the confirmation through gel electrophoresis and visualizing the gel from the gel dock or gel dock easy analyzer, we send the selected samples for the sequencing, while some of the selected samples were sent for histopathology or processed for histopathology. Now this is another study with the pathogenicity assay on white hegon layers. 2 groups were selected. One was the grower birds, one were the mature birds. The grower birds were of 18 weeks of age, while the mature birds of 44 weeks of age. So we inoculated the group B and D with our inoculum that we isolated from our field strains and noted different responses, and the parameters studied were as under. Now coming to our results parts, as I told you, 2,720 samples were processed and analyzed through RT-PCR. And on the images, on the right side, the images you can see 393 base pair, that is the partial S1 gene, the targeting at 393 base pair, while the below image, it's the full S1 gene that is the targeting at 1720 base pair. And from these 2720 samples, 625 samples came out positive. In case of layer, total 1090 samples were processed with a positive percentage of 26.60. But in case of broilers, 1630 samples were processed with a positive percentage of 20.55. Now, it's an interesting study we did that deduced amino acid similarity between our strains that we isolated, and the strains from the already submitted NCBI and the vaccinal strains that were currently the birds being immunized by those. The 491 and the H120 strains are currently being used for the vaccination of the bird that are highlighted in the red region. You can see there is a difference of 22 to 28% respectively of our field strains from the vaccinal strains. This means the vaccinal strains are only providing 72 to 78% protection to the birds. As I've told you in the introduction, that there are continuous emergence of antigenic variants and the coronavirus, positive sense, single-stranded. So that's the main cause that the mutations are occurring and the vaccination is a failure. Now, if we wanted to check our phylogenetic analysis that from where our strains belong, so the red ones highlighted are our two isolates, selected isolates from our study. These are the full S1 gene, and these belong to the genotype 1, lineage 24 of the Indian isolate. And this work has previously been done by Dr. Khalid Naeem Khwaja student Sabarfiq, in which her strain PK973 also belonged from the genotype 1, lineage 24. And this is the multiple sequence alignment. On the right side, you can see the dashes are indicating the missing amino acid gap. That means that there is a lot of difference between the field strain and the vaccinal strain currently being employed to vaccinize the birds. Now, this is the second part in which we did the pathogenicity assay in the white leghorn hens. In first part, on the upper left corner, the egg production percentage of infected mature layer birds were seen. At the week first, there was no significant change, means in the group C and D, the egg production was not changed. But from week 2 to week 12, egg production significantly decreased in the infected mature group D. On the right above side, egg weight gram of IBV infected mature layer birds, you can see from week 1 to week 12, the egg weight significantly decreased. Now, eggshell thickness of IBV infected mature layer birds, you can see from the week 1 and week 2, there was no significant change in the eggshell thickness size. Because in this disease, the eggshell thickness is decreased and the eggs become porous or soft-shelled eggs. But from week 3 to week 12, you can see a noticeable change in the eggshell thickness. In the in vivo phagocytic potential of macrophages, means the macrophages usually clear up the injected antigen. In this case, we injected with carbon ink. And you can see in group A, B, infected in the group A, B, and C, D, there was high time took a lot of time for the macrophages to clear up the antigen. Now, grossly, if we observe the birds, you can see water-filled cyst in the abdomen, and there is a kidney that are bulging out from the sockets and urea deposits. And in case of the D image, you can see the trachea is inflamed. And this is a proof in the histopathological images that you can see the photorenal parenchyma of the mature layered bird showing acute tubular necrosis with pycnotic nuclei, while in the right side, the trachea, you can see the slough of epithelium and necrotic changes shown by the black arrows. Now, significant findings and conclusions from the results of current project. It has been concluded that IBV classical and variant strains are present throughout our sampling area, that is the Faisalabad division. The unvaccinated birds showed high disease prevalence. Experimentally, it showed celiostasis. The molecular analysis of selected samples that already told you the 22-28% gap and difference in the production of currently available vaccines given to the birds. Now, this immunity gap may be the reason that the infection is spreading, but still we are doing a very high vaccination schedule, but still this infection is spreading. Now, pathogenicity assay in white leghorn birds, the egg weight were decreased to less than 43 grams, egg production were decreased to less than 26%, and macrophages were decreased as in the immunological parameter I have told you, and they were D-shaped and wrinkled eggs. Now, our future perspective is that our strain that we have isolated, the UAF sequence 9 session number is provided, can be chosen for the propagation of virus, and we can use this strain. I would like to thank the funding agency, PARC ALP, for funding all my PhD research work, and my technical knowledge that was provided round the clock by Dr. Khalid Naeem Khwaja and my supervisor, Dr. Muhammad Kashif Salimi. Thank you very much.
Thank you very much, Ahad. Our next speaker, Ms. Tahreem Ali. She is Research Associate, University of Central Punjab, and she will speak on molecular characterization and antibiotic resistance pattern of Clostridium perfringens type A isolated from necrotic enteritis positive birds. Tahreem, please, you have 10 minutes.
Assalamu alaikum. Starting with the name of Allah, who is most Rahman and Rahim. I am Tahreem Ali from University of Central Punjab, Lahore. Today the topic of my talk is molecular characterization and antibiotic resistant pattern of Clostridium perfringens type A, isolated from necrotic enteritis. Clostridium perfringens is a gram-positive, rod-shaped anaerobic bacteria. Normally, it is present in the intestinal tract of birds. Under certain conditions like predisposing factors, feeding of low-quality feed, long-term use of antibiotics, environmental stress, presence of other diseases, especially like coccidiosis, Clostridium perfringens cause ulcerative or necrotic enteritis, which will lead to the weight loss of birds, lower feed conversion ratio, and also the mortality reasons in the birds. The major potent and the lethal toxin in Clostridium perfringens is alpha toxin, which cause the necrotic enteritis in falls. Use of antibiotics in our daily life led to the antibiotic resistance in the birds, which is difficult to treat. We will talk about to all or study this. We designed a project. In that project, we isolate and characterize the Clostridium perfringens type A from the poultry birds having lesions of necrotic enteritis. For that, molecular confirmation was done. 16S ribosomal RNA and alpha toxin specific polymerase chain reaction was carried out. 2 qualitative assays were done: toxin detection by antigen specific ELISA, followed by toxin characterization by SDS page. At the end, antibiotic assay were analyzed using Kirby-Bart's method. On the basis of antibiotic assay, we analyze how much the antibiotic were resistant during this process. For that, 65 samples were collected from the intestine of the laying bird showing lesions of necrotic enteritis. These samples were supplemented on the reinforced Clostridia media and followed by Perfringens agar. Microscopic and the macroscopic characters were observed. Black, translucent, and the pure colonies with foul smell were observed, and these were suspected for biochemical conformation. For biochemical conformations, we performed it on sheep blood agar. On sheep blood agar, it showed double hemolysis. On 5% egg yolk agar, it shows the lecithinase enzyme activity. Catalase, oxidase tests were also performed. And different sugar fermentation tests were performed to check the Clostridium perfringens. Out of 65, 23 isolates were further processed and identified as Clostridium perfringens. Here it is the pictorial representation of pure growth of Clostridium perfringens. Microscopic characters, Clostridium perfringens showing double hemolysis on the blood agar. And the last one is the lecithinase activity on egg yolk agar. Then further 10 samples were processed to analyze on the molecular base. For that, DNA was extracted using commercially available DNA extraction kit. For that, QIAGEN or gene all were used. 16S ribosomal RNA amplification was done. Amplification was done and amplicons were sent to the lab for sequences. These sequences were further processed on NCBI BLAST. On the NCBI BLAST, we got the analysis and confirmed that it is Clostridium perfringens type A. Further phylogenetic analysis were performed. These sequences were submitted to the GenBank for accession numbers. These numbers were registered on the name of institute, and all were done by using MegaX tool on the phylogenetic analysis tree were showing with the word data bank analysis. Here is the alpha toxin gene amplification done using PCR, polymerase chain reaction. Toxin detection was done by using antigen-specific ELISA. Commercially available multi-screen ELISA kit was done. Sandwich ELISA was the procedure, and for that, we got 5 positive results. Alpha toxin showing 65.53%, 63.87%, 63.45%, 64.45%, and 37.75% were recorded. Further, toxin characterization by STS page was carried out. This is a vertical gel electrophoresis. On this, alpha toxin were detected at 48 kilodalton. Antibiotic assay were performed on the 5 isolates. These were all the antibiotics we used by using Kirby-Bor's disc diffusion method. Upon antibiotic sensitive assay, we recorded that Clostridium perfringens testis isolate were resistant to levofloxacin, ciprofloxacin, ampicillin, amoxicillin, doxycycline, oxytetracycline, while it was intermediate for tylosin, neomycin, polymyxin, and it was only sensitive to enrofloxacin. These all were compared with the available standard zone of inhibition on CLSI. To conclude, my talk is different molecular tests like toxin-specific PCR, antigen-specific ELISA, STS page techniques can be used to characterize pathogens. Bacterial diseases must be treated based upon antibiotic-sensitive pattern like in my studies, enrofloxacin was the choice of drug. Thank you so much. Jazakallah.
Okay, our next speaker is Ms. Masham Mukhtar from University of Veterinary and Animal Sciences, Lahore. She will speak on molecular and immunological characterization of markerless isogenic knockout mutants of Salmonella gallinarum as a potential candidate for attenuated vaccine in poultry.
Auzubillahiminash-shaytanir-rajim. Bismillahir-rahmanir-rahim. I am Masham Mukhtar and I am doing currently PhD under the supervision of Professor Dr. Amir Ghafoor Bajwa. And I am from University Diagnostic Laboratory, UAS, Lahore. And I am giving presentation on molecular and immunological characterization of markerless isogenic knockout mutant of Salmonella gallinarum as a potential candidate for a live attenuated vaccine against fowl type in poultry. I think you might bored with my long title topic, but I hope my results may cherish you at the end of the discussion, inshallah. So, लैपटॉप ठीक है चल नहीं रहा ना उसकी आगे ट्रांसमिशन नहीं चल रही मैडम इंट्रोडक्शन से करना है ना इंट्रोडक्शन से करूँगी इसको मैसेज So we identified the Salmonella gallinarum, which cause fall typhoid, results into 25 to 60% mortality in the infected flock. If we come into the economical impact, if a farmer is raising a breeder, then the cost of one bird is 3,700 rupees. If he has 1 lakh birds in his farm, then it roughly estimates 3,370 million rupees. If flock hit by Salmonella gallinarum infection at that time, the farmer has to sell out the birds as meat in 67.5 million. So, 302.5 million loss a farmer can face by a Salmonella gallinarum infection.
Sorry, we have a little change in the program because chief guest has also arrived. So we will continue this technical session after that small session. So up till now, the presentations, those are presented. I will request Professor Dr. Masood Akhtar to conclude first 5 presentations. यहाँ पे आ जाते हैं। वो बाद में कर लें।
Bismillahirrahmanirrahim. First of all, I would like to congratulate the organizers of the Poultry Science Conference for organizing such a wonderful event. Once again, thank you very much for all the organizers to provide me the opportunity to attend the conference and to conclude the session. The first session theme was the poultry health, and in this session there were total 11 presentations will be there in this session, and the first presentation that was the keynote by Professor Dr. A.D. Anjum, who's the authority in the poultry, and I don't have any words to express my sincere thanks to Dr. A.D. Anjum for such a wonderful presentation. Although due to the time limit, he concised the presentation. Anyhow, he gave us the overview on the poultry health indicators. Then he also discussed the diagnosis of the poultry diseases, and he presented the data regarding the main poultry diseases, especially the diseases which may cause the immunosuppression in the poultry. Then the second presentation, that was Hafiz Muhammad Suleiman. He was from University of Agriculture, Faisalabad, and he discussed the co-infection of chicken infection anemia virus with fowl adenovirus in commercial poultry, and he concluded from his results that the co-infection that exists between the chicken infectious anemia virus and the adenovirus, and he recommended that if we have to prevent the infection from the co-infection, we have to make measures, and he recommended for the vaccination program. Then the third presentation, that was on the avian influenza, that was the isolation of the avian influenza from the commercial layer facilities. It was the presenter was from the University of Veterinary and the Animal Sciences, and the presenter, they concluded that the environmental samples can be routinely collected from the poultry farm to avoid the infection of the avian influenza. Then the third presentation, that was on the molecular characterization of the Mycoplasma gallisepticum, and that presentation was from the College of Veterinary Sciences, the University of Agriculture, Peshawar, and it was concluded by the researchers that the Mycoplasma gallisepticum is the most prevalent pathogen in that area, I mean the KPK, and they also recommended that the disease, it is of systemic in nature, as they have observed the different lesions on the different organs of the birds. Then the next presenter was Maria Jamil. He was from the University of Agriculture, Faisalabad, and he discussed the immunopathological effects of avian adenovirus infection and the alpha toxin if if the birds are given the feed contaminated with the alpha toxin. And it was confirmed by the presenter in her study that the alpha toxin adulterated feed led to reduction in the bird performance, and it was also concluded that it has the immunosuppressive effect. These were all about the first 5 presentation which was presented in the poultry health session, and I can safely say that all the presenters, they did the excellent job, and I would also like to congratulate the organizers and the technical team who choose these presentation. All the presentations, these were international standard. Thank you very much. And the remaining presentation, these were concluded by Dr. Anas Sarwar Qureshi. Thank you very much.
Thank you very much, sir. Now this is a time for distribution of shields to our co-chair, chairs and speakers. I will request Dr. Hanif Nadeer Chaudhary, Chairman of the PSC and President WVP of Pakistan, to please come on the stage. I will request Professor Dr. Muhammad Akram Munir, sir, please come on the stage. I will request Professor Dr. Muhammad Athar, sir, please come on the stage. Professor Dr. Athar Mahmood. And also Dr. Athar from High Tech, sir, please come on the stage. And I will request Professor Dr. Khalid Naim Khwaja to please come on the stage to present the shields to our chairs, co-chairs and speakers. शील्ड रेडी है जी, शील्ड रेडी है आपकी, डॉक्टर साहब शील्ड रेडी है, First, I will request Professor Dr. Anas Sarwar Qureshi, chair of this session, to receive the shield from our honorable guest, Professor Dr. Anas Sarwar Qureshi. I will request Professor Dr. Masood Akhtar, co-chair of the session.
ये जो साहब अभी शील्ड दे रहे हैं, डॉक्टर खालिद नईम ख्वाजा साहब, इन्होंने इंटरनेशनल स्टैंडर्ड्स की डायग्नोस्टिक प्लस रिसर्च भी लेबॉरेटरी इस्लामाबाद में बड़ी हाई इन्वेस्टमेंट में बड़ी हाई क्वालिटी लैब बनाई है. सो क्लैपिंग फॉर हिम.
Now I will request Professor Dr. Sarzameen Khan, Dean Faculty of Veterinary and Animal Sciences, Agriculture, University Peshawar. I will request Professor Dr. Mazhar Ayaz from Chulistan University of Veterinary and Animal Sciences, Bahawalpur. Now I will request Professor Dr. Ahmadeen Anjam famous by the name of A.D. Anjum. Sir. I will request हाफिज़ मोहम्मद सलमान खालिद. मीनवाइल एक बात आपसे शेयर करना चाहता हूँ. हनीफ नदीर साहब यहाँ मौजूद हैं कि 2009 में मोरक्को में WP की कॉन्फ्रेंस पे प्रोफेसर हाफिज़ ने एक सेशन कंक्लूड किया, तो उसने उसमें दुनिया में पोल्ट्री की डेवलपमेंट दिखाई. तो पाकिस्तान से उसने 2 रेफरेंस शो किए, एक डॉक्टर मुकर्रब कुरैशी साहब का और एक डॉक्टर A.D. अंजम का एडिनो वायरस के हवाले से. दिस वॉज ऑनर फॉर अस. Next is Dr. Shahan Azim from University of Veterinary and Animal Sciences, Lahore. Dr. Muhammad Saeed from Agriculture University, Peshawar. Miss Maria Jameel from University of Agriculture, Faisalabad. Mr. Ahad Fiaz from University of Agriculture, Faisalabad. Ahad चेंज कर लेने के बाद मिस तहरीम अली फ्रॉम यूनिवर्सिटी ऑफ सेंट्रल पंजाब. Miss Farzeen. Dr. Abdul Rahman Ansari. Dr. Abdul Rahman from College of Veterinary and Animal Sciences, Jang. Miss Masha Mukhtar. मिस्टर उमर ज़हूर। ये प्रेजेंटेशन हम कंटिन्यू करेंगे उस सेशन के बाद, जो 4 रिमेनिंग प्रेजेंटेशन थे, वेरी इंपॉर्टेंट। सारे लोग यहाँ रहेंगे, हम इसके बाद उनको कंटिन्यू करेंगे। बाजवा साहब की बहुत इनपुट है इसमें।
नेक्स्ट है जी प्रोफेसर डॉक्टर मोहम्मद काशिफ सलीमी साहब। काशिफ सलीमी साहब भी किसी तारुफ के मोहताज नहीं है और पोल्ट्री इंडस्ट्री में इनका बहुत बड़ा कंट्रीब्यूशन है एज ए रिसर्च, और WVPA को लीडिंग जो है जनरल सेक्रेटरी में WVPA के पाकिस्तान में। सर ये शील्ड नेक्स्ट मैं दावत दूंगा काशिफ सलीमी साहब को कम बैक ऑन डाइस एंड से समथिंग अबाउट डॉक्टर अकरम मुनीर साहब। ऐसा भाई
अकरम मुनीर साहब को अभी देनी चाहिए। ओके जी, नेक्स्ट एक हमने अवार्ड है, एक्चुअली दिस इज़ हॉल ऑफ फेम ऑफ ऑनर, दैट इज़ वर्ल्ड वेटनरी पोल्ट्री एसोसिएशन WVPA ग्लोबली प्रेजेंट करती है। तो हमने ये पाकिस्तान ब्रांच से हनीफ नज़ीर साहब की एडवाइस पे स्टार्ट किया था। यस, तो इससे थोड़ा सा ब्रीफ मैं इसका कर दूं कि हॉल ऑफ फेम ऑफ ऑनर में जो टॉप नॉच साइंटिस्ट हैं ग्लोबली उनको दिया जाता है। जैसे लास्ट जो WVP की कॉन्फ्रेंस में डॉक्टर डेविड स्वेन को दिया गया था US से, तो हमने ये अवार्ड 2018 में फर्स्ट जो था वो डॉक्टर जरगाम साहब को दिया था, और सेकंड अवार्ड जो है वो हमने प्रोफेसर डॉक्टर अकरम मुनीर साहब के लिए अनाउंस किया। तो मेरी रिक्वेस्ट है कि ब्रीफली, ये बहुत इंपॉर्टेंट है जो अकरम मुनीर साहब की जो बायोग्राफी है, आई विल ट्राई टू कट शॉर्ट। डॉक्टर अकरम मुनीर ग्रेजुएटेड फ्राम यूनिवर्सिटी ऑफ पंजाब इन 1970 and he did his master from University of Agriculture Faisalabad in 1978, and he also did MS in veterinary virology from United States in the shortest period of 16 months. Then he did his PhD from University of Minnesota in 1987, and Professor Dr. Akram Munir has more than 100 publications, and he was also one of the key person in the Prime Minister task force for education. And he is well-renowned scientist for controlling the avian influenza in Pakistan, and he is a teacher of teachers. And currently, Professor Dr. Akram Munir is working as a chairman department of pathobiology, Rifa College of Veterinary Sciences, and he is also on the editorial board of Pakistan Veterinary Journal. So I will request Dr. Hanif Nazir to present Hall of Fame of Honor Award and certificate. And with your permission, I will also be part, because I am the secretary of WVPA Pakistan branch.
Kashif Salim sir. Kashif Salim sir, thank you very much. Even there is a lot more to say about Dr. Akram Munir sahab, his contributions for the poultry industry, for the, first it was College of Veterinary Sciences, then it became university. So he has a lot of contributed for the students, for the microbiology. And there is another award from the, one award is going from WVPA, and the second award, Lifetime Achievement Award, is going through Pakistan Poultry Association.
you very much, sir. We will continue this session after the... Okay, we have few words of wisdom from Professor Akram Munir.
Thank you very much. I am honored by PPA and World Poultry Science Association. I am really thankful to Dr. Hanif Nazir, Khalid Naim, Athar, and other. And there are many of my students sitting here. मेरी यही एक कोशिश रही है, मुझे पता है कि डॉक्टर A.D. अंजम साहब और डॉक्टर सिद्दीक मरहूम, हम तीनों का एक ही मिशन था कि पोल्ट्री डिजीज़ को डायग्नोज करना है, और इस सारे काम में हम हेल्प लेते थे डॉक्टर खालिद नईम ख्वाजा से। वो काम अल्हम्दुलिल्लाह आज भी ख्वाजा साहब ने कंटिन्यू किया हुआ है। आई हैड द ऑनर टू विजिट हिज न्यू लैब, जहाँ मेरा ख्याल है दुनिया जहाँ की जो भी कोई फैसिलिटी पोल्ट्री रिलेटेड हो या माइक्रोबायोलॉजी का डायग्नोस्टिक रिलेटेड हो, वो सारी वहाँ जमा है। तो आई एम रियली थैंकफुल, और मेरी ख्वाहिश है कि ये जो बीमारियाँ हम शुरू से सुनते आए हैं, इनका किसी तरह कला कमा किया जाए, कम से कम इतना कंट्रोल कर लिया जाए कि वो हमारे इकोनॉमिकल लॉसेज जो हैं वो कम से कम हो सकें पोल्ट्री को। थैंक यू वेरी मच।
Thank you very much, sir. Please have seats. और हम ये जी अतर साहब रनीफ साहब रुकते हैं, बाकी नीचे फ्रंट चेयर पे आ जाए आप, और अतर साहब रुकते हैं। और हम ये सेशन कंटिन्यू करेंगे। जो हमारे चेयर्स और को चेयर हैं वो सेशन में होंगे, जो 4 प्रेजेंटेशन रहती हैं।
Thank you very much for the participants. अच्छा जी, हमारे जो चीफ गेस्ट हैं, इज़ अबाउट टू अराइव विद इन 10 to 15 मिनट्स इंशाल्लाह। अभी इनॉग्रेशन के लिए वो हॉल में गए हैं, इसके बाद यहाँ पे आ रहे हैं। और आप सब दोस्तों से दरख्वास्त है कि कीप सिटिंग। और इसके साथ साथ एक छोटी सी अनाउंसमेंट और करता चलूँ जी, स्पेसिफिकली फॉर द स्टूडेंट्स। कल का दिन जो है हमारा एक, आज तो चलो जिस तरह स्टडी सर्कल था आपका एक लिहाज से, कल जो है यहाँ पे क्विज़ प्रोग्राम होगा, अम्यूज़मेंट होगी। वी नीड फुल पार्टिसिपेशन ऑफ द स्टूडेंट्स, जिसमें जनरल नॉलेज भी होगा, गिफ्ट हैम्पर्स होंगे, एंड दैट इज़ अ फुल डे फुल पैकेज प्रोग्राम। तो आई रिक्वेस्ट ऑल ऑफ यू गाइस टू पार्टिसिपेट। और अम्यूज़मेंट तो होगी, नॉलेज उसमें खासा होगा, लेकिन वो लेक्चर्स के थ्रू नहीं होगा, थ्रू 1-to-1 डिस्कशन इसके थ्रू होगा जी। अच्छा, मैंने रिक्वेस्ट की है डॉक्टर अतर साहब से कि मुझे ज़रा 2 मिनट चाहिए इनके, डॉक्टर हनीफ साहब के। हनीफ साहब प्लीज़ तशरीफ़ रखें, आपसे हम थोड़ा सा भी बात करते हैं अभी। लेकिन एक क्वेश्चन आया था जी हॉल से, वेरी इंटरेस्टिंग। ये नाम सही है या नहीं पता तो नहीं मुझे। सालार सिकंदर साहब हैं जी, यहाँ पे मौजूद हैं जी, सालार सिकंदर साहब। अच्छा, क्वेश्चन बड़ा जो है वो ज़बरदस्त है, और इसका आंसर जो है वो डॉक्टर अतर से बेहतर बंदा कोई नहीं है जो दे सके। क्वेश्चन ये जी कि पहले अंडा आया था या मुर्गी? तो आई फाउंड हिम टू द बेस्ट पर्सन टू टॉक टू। तो मैं दरख्वास्त करूंगा डॉक्टर अतर साहब से कि सर इसका प्लीज़ ज़रा आंसर कीजिएगा।
पहले ना अंडा आया ना पहले मुर्गी आई, सबसे पहले मुर्गा आया, क्योंकि उसने आगे टेंट लगाने थे। क्योंकि टेंट लगना था, फिर अंडे या मुर्गी का हिसाब चलना था। तो इसका जवाब यही है।
डॉक्टर साहब गए आपने मुर्गे का पूछा नहीं, लेकिन इसकी मज़ीद जेनेटिक्स में हम नहीं जाते, वो आप डॉक्टर साहब से यूनिवर्सिटी में रब्ता कर सकते हैं। और आई बिलीव कि जिस तरह हम डॉक्टर साहब से जो है मुस्तफ़ीद हुए हैं, 97-98 की बात है, तो उम्मीद है कि आप लोग आज उसी तरह से महसूस होते होंगे। लेक्चर में भी डॉक्टर साहब के मज़ीद खुश तो नहीं हुआ होगा सर, वैसे कि वैसे ही होगा लेक्चर अभी जैसा हमारे दौर में था। अच्छा, अब मैं एक छोटा सा जो ब्रेक आया है बीच में, सिर्फ हमारे गेस्ट की वजह से, इससे जो प्रेजेंटेशंस हैं, रिमेनिंग प्रेजेंटेशंस विल कंटिन्यू आफ्टर दिस सेशन, और बहुत इंपॉर्टेंट टॉपिक्स हैं। आई बिलीव कि अभी तक भी आपने बहुत एंजॉय किया होगा, और इन द कमिंग सेशन भी आप मज़ीद इससे मुस्तफ़ीद होंगे। खाना जो है जी, इसके बाद लेक्चर्स होंगे, उसके बाद क्वेश्चन आंसर सेशन होगा। वी हैव गॉट वेरी इंटरेस्टिंग क्वेश्चंस अदर दैन दिस क्वेश्चन, इट इज़ ऑल्सो इंटरेस्टिंग बाय द वे। तो उसमें उम्मीद है कि आप लोग पार्टिसिपेट करेंगे। और जो खाना है वो तकरीबन अराउंड 2:30 के होगा, तो हम 2:30 तक आपको ज़रूर फारिग करेंगे. सो आई बिलीव यू मस्ट बी हियर टिल 2:30. अब मैं रिक्वेस्ट करूँगा डॉक्टर हनीफ नज़ीर साहब से कि प्लीज़, उस वक्त हम टाइम थोड़ा था, ही हैज़ मोर लॉट टू से अबाउट दिस कॉन्फ्रेंस एंड द रिसर्च आर्टिकल्स, क्योंकि वायरस के ऊपर इनकी बहुत ज़्यादा स्टडीज़ हैं. सो आई वुड रिक्वेस्ट हिम कि थोड़ा सा मज़ीद हमें वायरसेस के बारे में और जो प्रेवलेंस है डिजीज़ के बारे में मज़ीद हमें एक्नॉलेज करें. डॉक्टर नज़ीर.
जी डॉक्टर साहब, बहुत बहुत शुक्रिया डॉक्टर शहजाद साहब, और आप लोगों का भी, जैसे उन्होंने प्लान दिया है कि बीच में जो ब्रेक है इसमें वो होगा इनागुरल आईपेक्स, और उसको भी आप एंजॉय करें. फिर रिमेनिंग पेपर्स होंगे, फिर उनकी कंक्लूजन होगी. तो जैसे इन्होंने कहा कि अभी गेस्ट आने में भी थोड़ा टाइम है, तो मैं आपको एक वंडरफुल, बड़ी अजीब बात आपको शायद लगेगी कि मुझे ग्रेजुएट किए हुए ऑलमोस्ट 42 इयर्स होने को हैं, और मैं महसूस करता हूँ, ये ज़रा गौर से सुनिएगा जो भी घूम रहे हैं, क्योंकि सरकारी नौकरियाँ अब मेरे ख्याल है बिल्कुल नहीं हैं या बहुत कम हैं, और आप लोग ज़्यादा हैं, तो आपने अपनी कॉम्पिटेंसी के हाईएस्ट लेवल पे अपने आप को रखेंगे तो आपको मौका मिलेगा. ये जो होता है ना, बंदा कहता है यार वो फलाना बंदा बड़ा जॉली है, बड़ा शरारती है, बड़ा ये है, किसी हद तक तो वो है, लेकिन यकीन मानिए कि जो आगे आपके लिए कंपटीशन है वो बहुत बहुत स्ट्रॉन्ग है. तो आप हर बात पे तवज्जो दें. क्या पता कि आपको कहाँ पे किस चीज़ की जो है वो हेल्प हो जाए अपना कैरियर बनाने के लिए. लेकिन फॉर श्योर ये जो इल डिसिप्लिन एटीट्यूड होता है या बच्चा टाइप फीलिंग होती है, इसका तो मैं श्योरली आपको बता सकता हूँ कि या तो अब्बा जी की आमदन पे आप गुजारा करेंगे, और या फिर आप किसी सरकारी नौकरी मिल जाए तो उसमें गुजारा करेंगे, जो कि ना होने के बराबर हैं. और अगर आपने अपना खुद कमा के खाना है तो लिसन एवरी वर्ड, और उसके अंदर कोई ना कोई आपको डायरेक्शन मिलेगी. आप देखें कि यहाँ किस कैलिबर के लोग इकट्ठे हुए हैं. अभी खालिद नईम ख्वाजा साहब को सुना, मुझे बता रहे थे कि लाइफ की जितनी सेविंग है वो उन्होंने अपनी ये डायग्नोस्टिक स्टेट ऑफ द आर्ट लेबॉर्ट्री डेवलप करने में लगाई है. और आप अंदाज़ा करें कि इससे बड़ी प्रोफेशनल कमिटमेंट क्या हो सकती है. तो जहाँ तक रही बात, मैंने एक दिन एक लालटेन देखी, और लालटेन आप लोग सब जानते होंगे, बत्ती भी उसे कहते थे. और वो जो घरों में एक छोटा सा वो दीवार के अंदर बना होता था, उसके अंदर वो लालटेन ज़्यादातर रखते थे. तो जो उस वक़्त लालटेन को साफ़ करता था घर का कोई यंगस्टर, तो उसके अंदर भी लालटेन साफ़ करने का एक स्किल होती थी. और वो स्किल जो है वो उसको मुनफरिद बनाती थी. और घर वाले वालिद साहब ये कहते थे कि फ़लाँ बच्चे को दें, ये बहुत अच्छा उस साहब. तो नेक्स्ट प्रेजेंटर कौन है जी, वो अनाउंस करेंगे. सो वी विल कंटिन्यू आवर प्रेजेंटेशन. आए जी डॉक्टर सलीमी साहब.
ओके जी ओके जी मशाम विल कंटिन्यू क्रॉस स्टेडियम पे सर में लग ओके
Assalamu alaikum. Sorry for the inconvenience. I am Misham Mukhtar and I am currently doing PhD under the supervision of Professor Dr. Amir Ghafoor Bajwa, and I am giving the presentation on molecular and immunological characterization of markerless isogenic knockout mutant of Salmonella gallinarum as a potential candidate for a live vaccine against fowl typhoid in poultry. So we identified the Salmonella gallinarum in a poultry which cause fowl typhoid, results in 25 to 60% mortality in the infected flock. If we come into the economical impact, if a farmer has a breeder stock in his farm, the cost of one breeder is 3,700 rupees. If he has 1 lakh birds in his farm, then roughly estimates is 370 million rupees. If at that time a flock hit by the Salmonella gallinarum infection, the farmer has to sell out all the birds in the sense of meat. So meat are sold out in 67.5 million rupees. So net loss 302.5 million a farmer can face by Salmonella gallinarum infection. So this huge economical loss needs to be addressed. So solution of the problem is vaccine. Vaccine may be killed or live attenuated. So live attenuated vaccine is a very right option. It should based on locobacillus. It should not revert back to the pathogen and should not have transovariant transmission. If we want to create a live attenuated gene modified vaccine, so we'll look into the genome of Salmonella gallinarum. we have picked two important genes. Number one, ARO gene. An aromatic amino acid helps in the growth of Salmonella gallinarum. If we target this gene will help the bacterial growth slows down and the bacteria will be available for the immune system. And the second one gene is SPI2. The Salmonella pathogenicity island type 2 plays an important role in the pathogenicity of Salmonella gallinarum infections. We believe that by deleting these two genes, Salmonella gallinarum ability to cause disease can be finished and will be minimized. By making such hypothesis, we use lambda red technology to delete these two genes. In this technology, we join antibiotic acid with start part and the end part of the gene to form a chimeric gene sequence. The gene of interest is replaced with chimeric gene sequence by homologous recombination. So antibiotic acid inserted into the chromosome in the replacement of gene of interest. Later on, antibiotic acid was removed by a technology, leaving a scar behind in the genome. So the gene is no more functional for its purpose. So the vaccine which has ability to trigger the immune response in the body but do not have ability to cause disease, this type of vaccine is called live attenuated gene modified vaccine. So once the vaccine is developed, some experiments were performed to check whether our vaccine creates a humoral immunity or cell-mediated immunity, or the birds will able to withstand the Salmonella gallinarum infections. So some experiments were performed. We were quite worried that will our mutant strain be able to withstand all these experiments, but thanks God we got a very promising results. So let's walk through you the selected results. As you can see, first of all we check the safety of the vaccine. In the first graph, the control group and the group which were vaccinated with our novel mutant strain showed a proper growth on the body of the birds after vaccination, while the other group which were infected with wild type bacteria survived birds showed not a proper growth but significantly less as compared to the other two groups. So next graph showed survivability of the birds after vaccination. Control group and the group which were vaccinated with mutant strain showed 100% survivability of the birds, but the birds which were infected with wild type bacteria, only 20% birds were survived. In the third one graph, we did depression scoring after the vaccination. Depression scoring in the control birds showing very high, but the control group and the group which were vaccinated with the mutant strain showed non-significant and a very few depression, as you can see on the graph. So we concluded here that there is no negative impact of the vaccine on birds. So how long bacteria stayed in the body of the birds, and our immune response can remove the bacteria from the body. The blue line showed that the birds which were infected with wild type bacteria, they remained in the body until end, while the vaccinal strain is removed by the immune system from the body within 12 to 14 days. This is an important feature of live attenuated vaccine, that it doesn't persist in the organs. So you can see the same results as shown in the liver graph. So next slide comes up with the immune response. What positive control a vaccine can play. Humoral immune response was measured at different time intervals by group D ELISA kit. Humoral immune response was highly increased in experimental birds as compared to other control group. Similarly, cellular immune system was also highly increased in experimental birds as compared to the control group. Cell-mediated immunity was measured by chicken interferon gamma kit. This is one of the most important data set. This data set showed that The birds which were challenged with Salmonella gallinarum infections, vaccinated birds remained unaffected, but the control group showed 80% mortality. We did Leon scoring in liver and spleen. We showed that the control group showing highest liver and spleen scoring, while the birds which were vaccinated with the mutant strain showed non-significant and a very few lesions in spleen and liver, as you can see on the screen. And the scoring also mentioned on the graph. So, crux of the story is money. We have estimated the loss of poultry by the Salmonella gallinarum infections. For example, if a population of broiler is 1020 million, the prevalence of broiler due to prevalence of Salmonella gallinarum due to ST infections 5.35%, and the mortality rate due to ST infections 25%. So the number of birds will be 13.64 million, and the money economical loss annually due to ST infections in the broiler will be 682.25. So similarly in the case of broiler breeder and layer. So at the end we have estimated total loss which will 9961.26 million rupees. So our promising results of our vaccine show that there is a hope we can save 10 billion rupees minimum annually by overcoming this Salmonella gallinarum infection. So thank you so much for your attention.
Next, madam. Thank you, madam. Our next speaker is Dr. Farzeen from University of Veterinary and Animal Sciences. Dr. Farzeen is here. Farzeen, please. She will speak. नहीं आ गई मैडम उनको आने दें ठीक नहीं ये मैडम आ गई पहले Farzeen will be speaking on the tissue tropism and molecular detection of BP2 gene of IBD in experimentally infected broilers.
Bismillahirrahmanirrahim and Assalamu alaikum. I am Farzeen Shafiq and my topic of presentation is Study on Tissue Tropism and Molecular Diagnosis of Infectious Bursal Disease Virus in Broiler Chickens. As you know, poultry industry plays a vital role in the economy of the country. The main threats which are faced by the industry is in the form of infectious and non-infectious disease. Among infectious ones, viral diseases are more common. The infectious bursal disease virus continue to be a serious problem mainly due to two main reasons: heavy mortality in young chickens and immunosuppression. The objective of this project is to identify the infectious bursal disease virus from field, to study the pathogenesis and tissue distribution of isolated virus in commercial chickens, to confirm the antigen in infected tissues, to compare the efficacy of commercial IBD vaccines. Methodology: experiment number 1, isolation and identification of IBD virus from field samples. For this purpose, we collected 100 bursal samples and processed for virus isolation. Molecular detection and histopathological studies. These are the results of the first experiment. The first picture shows the RT-PCR banks which are positive. The second picture shows the DNA sequencing and analysis along with the phylogenetic analysis. Experiment number 2, pathogenesis of field infectious bursal disease virus. For this purpose, we reared 200 poultry chicks in 4 different groups, A, B, C, D. The group A and B was infected with the virus and group C and D were the control groups. Different parameters were also evaluated, including clinical observations, gross pathology, histopathology, IHC, and serum biochemistry. Results of the second experiment shows the normal bursa of Fabricius, edema and hemorrhage, swollen bursal folds, pinpoint hemorrhages on thymus was also observed. Hemorrhages on pectoral muscles and congested and shrunken lumen was also revealed. Severe hemorrhages on bursal folds and bursal atrophy observed in group B only. Histopathological findings shows that hemorrhages and mild lymphocytic depletion in thymus, severe hemorrhages and leukocytic infiltration in urinary tubules, normal lymphoid follicle with normal epithelium, severe hemorrhages and lymphocytic depletion in bursa, lymphocytic depletion and leukocytic infiltration in bursa, lymphocytic depletion at bursal follicles. RT-PCR shows the positive results as you can see in the picture. Experiment number 3, comparative efficacy of commercial IBD vaccines against field isolates. For this purpose, we also reared 200 chicks in 4 different groups, A, B, C, D. Here, the group A was vaccinated with immunocomplex vaccine, the group B was vaccinated with the intermediate vaccine, the group C was vaccinated with the intermediate plus vaccine, and the group D was a control group. Different factors was evaluated, which include weight gain, bursa to body weight ratio, bursometry, ELISA, gross and histopathological lesions scoring. The results of experiment number 3 showed that there is no significant change in the weight gain, and ELISA antibody titer also showed very significant, very little results, positive results. These are the results of the bursometry. Only group D shows the significant difference in the bursometer. This is the bursometer which I have used during the experiment. Bursa to body weight ratio. This comparative chart shows the very little difference only in the group D. Gross lesions in the different groups were also observed. The results showed that isolated field IBDV VP2 gene sequencing revealed virulent strain existed. IHC's better diagnostic test to understand the mode of action of the recent IBDV field isolates towards the tissue tropism. Intermediate immune complex vaccines have better results than the other vaccines, namely intermediate plus, as they cause less damage to the bursa of the Fabricius with protective antibody titer. These are the research articles which I published recently, and thanks.
Thank you very much. Now our next speaker is Mr. Umar Zahoor, He is an MPhil scholar in University of Veterinary and Animal Sciences, PhD scholar. And he will be speaking on phylogenetic and cross-immunization analysis of field isolates of IBV from various regions of Pakistan. Umar, please.
मैं साथ साथ आने वाले जो हमारे मेहमान हैं उनका शुक्रिया भी अदा करता चलूँ, ख़ुसूसी तौर पे राय मनसब अली करल साहब, चौधरी अजमत साहब, खालिद सलीम मालिक साहब, राना गुलाम मुस्तफ़ा साहब और हमारे कुछ दोस्त जो सिंध से तशरीफ़ लाए हैं। सिंध में इस वक्त बहुत सारे मसाइल हैं, उसके बावजूद उन्हें टाइम निकाला। कमाल अख्तर सिद्दीकी साहब, हमज़ा साहब, अली साहब यहाँ पे तशरीफ़ लाए और उम्मीद है कि 2 से 3 दिन जो हमारी ये एक्सपो चलेगी इसमें इंशाल्लाह सैर हासिल यहाँ से इस्तिफ़ादा भी करेंगे। थैंक यू वेरी मच सर।
Bismillahirrahmanirrahim. My name is Umar bin Zahoor. Currently, I am doing PhD in microbiology under the supervision of Professor Dr. Amir Ghafoor Bajwa in UDL, US, Lahore. The topic of my presentation is phylogenetic and cross-immunization analysis of field isolates of infectious bronchitis virus from the various regions of Pakistan. You can see, for those who don't know, infectious bronchitis virus is the coronavirus of the poultry. We are quite lucky that infectious coronavirus didn't cause that much extent of damage to humans as we are expecting in the poultry in Pakistan. So we identified the problem and its intensity and we've tried to find the solution for it. If we talk about the problem of the infectious bronchitis virus, so we have seen that despite of vaccination, the field outbreaks currently occur in the poultry in Pakistan. And there is very less work reported about the characterization and isolation of the infectious